Imagine getting medicine that isn't just based on, your symptoms, but is designed specifically for you,
Hmm.
tailored to your unique biology, your genes, the proteins your cells are making, maybe even the specific environment inside a tumor.
Right.
Think about getting the right treatment the very first time, maybe skipping therapies that wouldn't work anyway, and avoiding those harsh, unnecessary side effects.
Yeah, that vision of truly personalized healthcare, what we call precision medicine, it feels like it's coming into focus much faster now.
Yeah.
And frankly, artificial intelligence is really the essential tool making it possible.
Exactly. And that's really the core of this deep dive today. We're going to explore precision medicine, focusing specifically on how AI is helping us navigate this, well, mind-boggling complexity of biological data, from the fundamental code of our DNA all the way down to the microscopic world inside and around our cells.
It's a lot to unpack.
It is. So our goal is to understand how this is transforming the fight against major diseases, particularly cancer, but also with implications for urgent situations like emergency care, too.
And to help us sort of chart this course, we've pulled together some really compelling sources. We've got a detailed examination of how AI is impacting cancer treatment, especially how it's helping us understand the tumor's interaction with the immune system,
Okay.
and also how it's revolutionizing how we analyze medical images. Then we also have a look at what goes into, say, a national precision medicine strategy.
Right, the bigger picture.
Yeah, and even a glimpse of how this approach could speed up diagnoses in critical, time-sensitive situations, like certain neurologic emergencies.
Okay, so our mission for you in this deep dive is to unpack how AI is crunching these absolutely massive data sets, genetic sequences, protein profiles, detailed medical scans, even digital microscope slides, all to help researchers and doctors discover better ways to identify diseases, predict which treatments will work best for you, and ultimately just make healthcare far more precise and hopefully effective.
Okay, so let's unpack this idea of precision medicine first.
Right.
At its heart, it's really about saying goodbye to the old, one-size-fits-all model.
Right.
It's about tailoring medical treatment to the individual patient,
Yeah.
taking into account their unique makeup, not just their symptoms.
And I think it's easy to hear precision medicine and immediately think of things like targeted cancer drugs, which, yeah, that's a huge part of it. And that's where a lot of the AI development is happening right now.
Absolutely.
But our sources really highlight that the principles are much broader. That mention of a neurologic emergencies like maybe a stroke, minutes matter there.
Critically.
So if AI can rapidly analyze a head CT scan and flag something maybe a human eye might take longer to spot, well, that's precision medicine in action too, right? For a critical, time-sensitive situation.
Absolutely. And the fuel for this whole approach, really, is data. Specifically, these vast quantities of incredibly detailed biological data that we often group under the umbrella term omics.
Ah, the omics revolution. Sounds a bit technical maybe, but it's really just breaking down biology into these different layers of information.
Exactly. It sort of starts with genomics, the study of our DNA or genetic blueprint. You remember the Human Genome Project?
Oh yeah, huge undertaking.
Sequencing that first human genome was this massive multi-year global effort. Hundreds of labs involved.
It's a monumental achievement at the time, definitely.
Completely. But now, with technologies like next-generation sequencing, NGS, sequencing a genome, is dramatically faster
Yeah.
and cheaper. But here's the catch.
Uh-oh.
It produces truly astronomical amounts of data. We're talking petabytes, trying to find meaningful patterns in that deluge of genetic code without AI. It's practically impossible.
Okay, so genomics is the blueprint, but what about what the cells are actually doing right now? What instructions are they following?
All right, that takes us to transcriptomics, studying RNA, specifically messenger RNA or mRNA.
Okay.
Think of mRNA as the working copies of the blueprint instructions that cells are actively using at that moment. It tells us which genes are turned on or being expressed.
Ah, okay, like seeing which specific parts of the building plans are being used on the construction site today.
That's a great analogy, yeah. Techniques like microarray and RNA-seq are used here. RNA-seq is generally more advanced. It can discover new RNA sequences, works with smaller samples.
Yeah.
But microarray is currently more cost-effective if you're just looking at known sequences. Analyzing this data helps us understand disease processes and how things like drugs are affecting gene activity.
And we can go even further down the chain, right? To the actual machinery the cells use, the things carrying out those instructions.
Yes, absolutely. That gets us to proteomics, the study of proteins, their structures, their functions. They're the workhorses, really.
Okay.
And then even deeper, you have metabolomics, which looks
Yeah.
at metabolites. These are the small molecules that are the products and intermediates of cellular metabolism.
So like the chemical fingerprints of what the cell has actually been up to.
Precisely. Metabolomics is a really promising field, though still emerging, for finding new biomarkers.
Yeah.
But analyzing the huge range of concentrations these molecules exist in, that's a significant challenge.
Wow. So you have these incredibly complex different layers of biological data, DNA, RNA, proteins, metabolites. Each one is massive on its own. How do you even begin to connect those dots and understand how they all interact?
And that's the critical role for AI. It's simply indispensable for integrating these diverse omics data types.
Okay.
AI algorithms can find patterns and correlations across these different layers, things we just couldn't see before. It gives a much more holistic view of what's happening in a patient's biology compared to looking at any single layer alone.
Hmm.
That's a key insight from our sources here. AI isn't just analyzing each dataset in isolation, it's integrating them to build that bigger, more complete picture.
And our sources make a point that it's not just the molecular biology, is it? Other factors are just as crucial for true precision.
Absolutely. That's so important. Precision medicine isn't purely about the molecular level.
Yeah.
Physiological factors like age, sex, ethnicity, even the stage of a disease,
Yeah.
they dramatically influence how a disease progresses and how a patient responds to treatment.
Right, like how a drug gets absorbed, distributed, metabolized, eliminated, the whole ADME process.
Exactly, ADME. And we can't forget lifestyle habits, either diet, smoking history, exercise levels. These have a profound impact and really need to be layered into the analysis for truly personalized treatment plans.
Okay, so we have all this complex data, biological and otherwise, and AI to help integrate and understand it. Let's maybe focus back on cancer for a bit, because that's where some of the most striking progress seems to be happening. Our sources talk a lot about the tumor immune microenvironment. What exactly is that, and why is it so important?
Right. The tumor immune microenvironment, or TIME, it's essentially the neighborhood surrounding a tumor. It's not just the cancer cells themselves. It includes all the other cells mixed in there. Immune cells, blood vessels, connective tissue cells, fibroblasts.
The whole ecosystem, basically.
Exactly, the ecosystem.
Yeah.
Understanding this complex environment is absolutely critical because it heavily influences how the tumor grows and, really importantly, how it might respond to therapies, especially immunotherapies that try to unleash the body's own immune system against the cancer.
Hmm.
The core insight here is that the composition and the organization of all those cells around the tumor can be just as important, sometimes more important, than the tumor cells themselves in predicting outcomes.
Okay. And the sources break down the time into different categories based on how the immune cells are interacting with the tumor. Is that right?
They do. Generally, there are three main types described. First, you have the infiltrated excluded, or i.e. times. These are often called cold tumors.
Cold tumors. Yep.
Immune cells, particularly T cells, which are the main cancer fighters, are present, but they're sort of stuck. They're in the tissue around the tumor or maybe trapped in fibrous barriers, but they're not actually getting into the tumor mass to attack it effectively.
Ah, so they're like soldiers outside the city walls, unable to get in and actually fight the battle inside.
Precisely. And as you'd expect, these cold tumors generally don't respond very well to standard immunotherapies, like immune checkpoint inhibitors or ICI.
Makes sense. So what's the opposite?
That would be the infiltrated inflamed, or I times often called hot tumor.
Hot tumors.
Right. These have high levels of immune cells, especially those T cells, infiltrating deep within the tumor itself. There's active immune signaling happening. They often express markers like PD-1, PD-L1.
Which we'll get back to, I'm sure.
Yes. And these tumors are much more likely to respond positively to ICI therapy.
Okay, so cold is less likely to respond, hot is more likely. What's the third type mentioned?
The third category is tertiary lymphoid structures, or TLS.
TLS. Okay.
These are interesting. They're structures that form within or near the tumor tissue itself, kind of like mini improvised lymph nodes right there at the site. They contain various immune cells.
So they can help or hinder.
Exactly. Depending on their specific cellular makeup and activity, they can either help generate effective anti-tumor immune responses, or, in some cases, they might actually suppress the immune attack. It's complex.
So just knowing the location and the type of immune cells in that neighborhood around the tumor gives you really crucial information about prognosis and treatment response.
It absolutely does. Changes in the time composition are frequently associated with worse prognosis. Our sources give some specific examples. Like in breast cancer tissue, compared to normal tissue, there are often more white blood cells overall, but importantly, a lower ratio of the killer CD8 plus T cells to the helper CD4 plus T cells within that mix.
Ah, so the balance matters.
The balance is key. Another example is cholangiocarcinoma. That's a type of bile duct cancer. The sources note that patients who tend to have better long-term survival often have relatively higher densities of CD8 plus T cells and B cells, and lower densities of regulatory T cells, TREGs, and suppressive M2 macrophages in their tumor microenvironment.
Wow.
Just knowing that blend of cells gives powerful predictive value.
Okay, and this is where AI really becomes essential, right? Trying to find those subtle, complex patterns in this immune landscape using all that omics data.
Absolutely. AI is critical in what we call immunogenomics. We use AI to analyze genetic and transcriptomic data specifically related to the immune system. It helps us find patterns associated with specific immune responses or disease states, effectively discovering new biomarkers that relate directly to the time.
And there are specific bioinformatics tools that AI uses for this kind of analysis.
Yes, the sources mention tools like CyberSort, for example. They're designed to take complex bulk omics data from a whole tumor sample.
Right, where everything's mixed together.
Exactly. And computationally deconstruct it to estimate the proportions of different immune cell types present, even closely related ones. The sources highlight its advantages in handling noisy data and its visualization capabilities, which are helpful.
What about looking even more closely, like at individual cells within that mix?
Yeah, that's where single-cell RNA sequencing, or cRNA-seq, comes in. It's a much more high-resolution technique that analyzes the RNA from individual cells within a sample.
Wow, okay.
This is hugely powerful because it can reveal hidden diversity within cell populations that you totally miss with bulk sequencing. It can even help identify rare cell types that might be playing a really critical role in how a tumor develops or responds to treatment.
That sounds like it provides incredible detail. And you also mentioned DNA methylation analysis. How does that fit in?
Right, DNA methylation is another important piece of the puzzle. Unlike RNA, which can be less stable, DNA itself is chemically very stable. Analyzing DNA methylation patterns, these chemical tags on the DNA, offers a potentially more robust and stable way to characterize the time over time.
I see.
Tools like CyberSort, also mentioned in the sources, use these methylation patterns to accurately estimate tumor composition. They can help classify tumors into those hot or cold types we talked about, and even identify patient groups with different prognoses based purely on these epigenetic patterns.
So we're using AI to dig deep into the biological code, the RNA messages, the methylation patterns, all to understand the immune cells around the tumor. But AI can also work with visual data, right? Like reading medical scans.
Yes, absolutely. And this is a huge area. AI in radiomics is a major and crucially non-invasive avenue for precision medicine.
Non-invasive is key.
Definitely. It involves using AI to analyze standard medical images, things like CT scans, PT-CT scan, MRI scans to predict things about the tumor and the patient's likely response without needing a tissue biopsy.
How does it do that?
AI algorithms are trained to look for incredibly complex patterns and textures within the image data, things often invisible to the human eye. These patterns are sometimes called radiomics signatures.
And this connects back to predicting how a patient might respond to immunotherapy, maybe based on those biomarkers you mentioned earlier?
Exactly. This ties directly into those important immunotherapy biomarkers like PD-L1 and tumor mutational burden, or TMB.
Okay, remind us quickly about those.
Sure. PD-L1 is a protein often found on cancer cells, or sometimes on nearby immune cells. It basically acts like a brake or a don't eat me signal to the immune system telling T cells to back off.
Right.
So drugs that block this PD-L1 interaction, the ICIs, help release that brake and unleash the immune response. And as our source points out, PD-L1 expression level is currently the only FDA-approved biomarker for selecting which patients get ICI therapy in certain types of lung cancer.
Got it. And TMB.
TMB, tumor mutational burden, is basically just the total number of genetic mutations within a tumor's DNA.
Okay. More mutations means?
Generally, a high TMB means the tumor is producing more abnormal proteins, called neoantigens. These neoantigens act like foreign flags that the immune system is more likely to recognize and target. So tumors with high TMB often respond better to immunotherapy.
And AI can predict these things, PD-L1 status, TMB levels, just from looking at a scan. That still feels almost futuristic.
It's really happening now. Our sources highlight studies where PTCT radiomics using AI analysis could predict PD-L1 status with reported high sensitivity and specificity.
Wow.
Other examples include using machine learning-based radiomics to predict those time phenotypes, the hot versus cold tumors, and forecast response to anti-PD-1, PD-L1 therapy in breast cancer. It can even predict the levels of those crucial CD8 plus fighter T cells and overall ICI outcomes, all from analyzing the scans.
So potentially, instead of needing an invasive biopsy just to test for PDL1 or TMB, you might be able to get a good prediction using AI on a routine scan.
That's the promise, yes. And it's not limited to just one type of image or one type of data either. Researchers are using multimodal deep learning,
Meaning,
meaning combining different types of data. For instance, using CT scan image features plus clinical data like patient history to build even more accurate models for predicting treatment response, say in gastric cancer.
okay, so AI is analyzing genes, RNA, proteins, metabolites, medical images. What about looking at the actual tissue slides that pathologists examine under a microscope, pathomics?
Yes, AI in pathomics is another really key area. This involves using AI to analyze digital versions of those glass microscope slides from tissue biopsies.
So digitizing the slides first.
Exactly. Then AI algorithms extract thousands of quantitative features from these images, things like the exact size and shape of individual cells, the texture of the tissue, how cells are arranged spatially relative to each other, details that are incredibly hard or impossible for a human pathologist to quantify precisely and consistently across a whole slide.
And then machine learning gets applied to all those visual features.
Precisely. Machine learning algorithms analyze these extracted features, identify the most important ones, find patterns, and build predictive models. These models can then be used for things like predicting a patient's prognosis or how likely they are to respond to a specific treatment based directly on the visual characteristics of their tissue sample.
This all sounds incredibly powerful, really transformative potential, but, moving something this complex from the research lab into routine patient care, that must face some huge obstacles, right?
Oh, absolutely. There are significant hurdles. One of the most fundamental, maybe even before you get to the fancy AI analysis, is simply managing the data itself.
Right, you mentioned petabytes of data earlier. That's just hard to even picture the
It
scale.
is. The scale of omics data is truly staggering. The Cancer Genome Atlas Project, TCGA, that alone is about 2.5 petabytes. By 2013, the European Bioinformatics Institute already held over 20 petabytes.
Wow.
Just storing this data, keeping it organized, secure, and importantly, accessible for researchers and clinicians globally is a massive, massive undertaking.
Well, some things like cloud computing are pretty essential for handling that volume.
They help immensely. Cloud platforms like Amazon Web Services are used by major initiatives like the NIH Human Microbiome Project, but the sources really underscore that the challenge isn't just storage capacity, it's making sure these massive databases are well curated and user-friendly, meaning the data has to be clean, standardized, annotated properly, and easy for people to actually find and use. If the data quality isn't there, or if it's too hard to access and integrate, even the most sophisticated AI algorithms can't effectively translate those potential breakthroughs into actual clinical practice. Garbage in, garbage out, basically.
Okay, so data management, curation, accessibility, that's a huge challenge.
Yeah.
What about getting these new AI-driven tests and therapies through the necessary approvals? The regulatory landscape sounds like it could be pretty complicated.
It really is. That's another major bottleneck. Precision medicine products often involve both a therapy and a diagnostic test. Think of a drug paired with a test to see who should get it.
Right.
So the question becomes, is it regulated as a drug, as a medical device, as both? This means they can fall into complex, sometimes unclear regulatory pathways.
But there's been progress.
There has. The good news is that the regulatory environment is adapting, maybe not always as fast as the science, but it is changing. This has been partly spurred by big government initiatives like the Precision Medicine Initiative back in 2015 and the 21st Century Cures Act in 2016.
And those legislative efforts have actually accelerated things on the approval front.
It seems so. The data shows a real shift. In 2005, only about 5% of newly approved drugs by the FDA were considered precision medicine products. By 2017, that number had jumped to over 30%.
That's a big jump.
It's a significant acceleration in just over a decade, suggesting the system is responding.
Can you clarify some of those regulatory complexities? Like, what are companion diagnostics?
Sure. Companion diagnostics, or CDX, are tests specifically designed and approved to be paired with a particular drug. They help doctors decide if a specific patient is likely to benefit from that therapy or perhaps if they're at higher risk of side effects. The classic example mentioned in the sources is the Herceptin drug for certain breast cancers and the Herceptis diagnostic used to identify patients whose tumors overexpress the HER2 protein. CDX tests are generally regulated as medical devices.
And the FDA prefers they're developed together.
Yes, the FDA actually recommends that companies develop and submit the drug and its companion diagnostic for approval together, ideally, to try and avoid delays in getting both to patients simultaneously. The source notes that even a small delay between the drug and test approval, as happened initially with Herceptin Herceptest, was seen as a potential risk.
Okay. And then there's this difference between commercially available test kits and tests developed by a specific lab, LDTs.
Yes, and this is where some significant confusion and debate has arisen, especially impacting precision medicine. You have IVD kits, in vitro diagnostic kits, which are commercially available tests developed by manufacturers. These fall under direct FDA oversight. Then you have laboratory-developed tests, or LDTs. Historically, these were tests designed, manufactured, and used within a single laboratory. Think of a test developed at a major hospital lab for its own patients.
Okay.
Traditionally, the FDA exercised what's called enforcement discretion for LDTs, meaning they didn't actively regulate them as medical devices. They were primarily overseen for quality standards under CLIA regulations by CMS, the Centers for Medicare and Medicaid Services.
But modern LDTs are much more complex now, using AI and genomics.
Exactly. Today's LDTs, particularly in precision medicine, are often incredibly complex. They might involve sophisticated next-generation sequencing, complex algorithms, maybe even AI analysis. They carry potential risks and benefits very similar to the commercially available IVD kits that are regulated by the FDA.
So the lines are blurring.
The lines are blurring, and this has led the FDA to occasionally step in and assert jurisdiction over certain high-risk LDTs. The MammaPrint breast cancer recurrence test is cited as an example where this happened, causing uncertainty in the field about which rules apply when.
That kind of regulatory uncertainty must make it harder for companies developing these tests to invest confidently, right?
It absolutely does. If you don't know what the regulatory pathway or requirements will be, it creates risk and can stifle innovation and investment. It really highlights the ongoing need for clear communication, maybe updated regulations that can adapt to the rapid pace of scientific innovation while still ensuring patient safety and test reliability.
Okay, so we've got data challenges, we've got regulatory complexities. What about the economics? Does this highly personalized, potentially more complex approach actually save money in the long run, or is it inherently just going to be more expensive?
That's the multi-billion-dollar question, isn't it? It's a crucial debate with points on both sides. There's significant potential for saving money.
That's so.
Well, one major way is by making clinical trials more efficient. If you can use a diagnostic test to identify patients who are most likely to respond to a new drug before the trial starts.
You need fewer patients?
Exactly. You can run smaller, more focused trials. And clinical trials are unbelievably expensive.
Right.
Our source gives some compelling stats here. Targeted non-small cell lung cancer trials using biomarkers had about a 62% success rate getting through phase I development. Compare that to only about 11% success for phase I trials overall.
Wow, big difference.
Huge difference. This improved success rate and the smaller patient pool needed are estimated to cut drug development costs significantly. The examples given are around a 26% cost reduction for those NSCLC trials and maybe 27% for HER2 positive breast cancer trials. That's serious money.
Okay, so savings in drug development. What about for the patient or the healthcare system directly?
Well, think about avoiding treatments that simply won't work for a particular patient. That saves the cost of the drug itself, the administration costs, and crucially, it helps patients avoid potentially debilitating side effects.
Right, the cost of managing side effects can be huge too.
Definitely. And the source points out the broader cost of adverse drug reactions estimated at over $30 billion annually in the U.S. back in 2013. If precision medicine helps stratify patients away from drugs that would likely harm them or just be ineffective for them, that's a direct cost saving for the healthcare system and, of course, much better for the patient's quality of life.
But there are economic challenges or counterarguments.
Yes, there are. One major concern is on the pricing side. If a new therapy targets a very small, specific patient population defined by a biomarker.
The market is smaller.
Right. Pharmaceutical companies might argue they need to price that therapy very high to recoup their substantial R&D costs over fewer prescriptions. This requires market pricing structures that can somehow accommodate that, which is tricky. Also, this whole stratification approach really relies on having a very good understanding of the underlying disease mechanisms so you can effectively identify those who will benefit. That's still a major challenge for highly complex conditions, like
Yeah.
many psychiatric disorders, for example.
Makes sense. And then there's the issue of getting insurance companies, the payers, to actually reimburse for all these sophisticated tests, right?
Right, that's probably one of the biggest barriers to widespread adoption right now. Payers, both private insurers and government programs like Medicare, are often hesitant to cover broad, comprehensive genomic testing or other advanced diagnostics without really robust outcome data.
They want proof it works and saves money or is worth the cost.
Exactly. They need strong evidence showing it definitively improves patient outcomes and is cost-effective overall compared to standard care. However, we are seeing some important progress. The source mentions an agreement between United Healthcare, a major insurer, and Foundation Medicine to cover comprehensive genomic profiling for certain lung cancer patients. That's a significant step.
And decisions made by Medicare are probably the most impactful overall in the U.S.
Without a doubt. Medicare is the single largest payer in the U.S. healthcare system, and its coverage policies often set the benchmark that private insurers tend to follow. So, widespread adoption of many of these sophisticated precision medicine tests and therapies will likely depend very heavily on how Medicare policy evolves in the coming years.
So, okay, despite all these challenges, data, regulation, economics, payer adoption, we are seeing real-world progress. Can you give us maybe a couple of tangible examples from the sources that show this, moving beyond just the research papers?
Absolutely. Looking at a broader strategic level, Singapore, for example, has launched a major national precision medicine initiative called PRECISE.
Okay.
Their goals are ambitious. They include things like building a comprehensive reference genome specifically tailored to their diverse population, developing concrete clinical pathways for how to actually use and return genetic results for important conditions like hereditary cancers or high cholesterol, and actively working to stimulate industry interest and investment in the precision medicine field within Singapore.
So really building the infrastructure and demonstrating the practical clinical value proposition.
Exactly. It's a national commitment.
Yeah.
And then circling back to that California example we touched on earlier, the potential for rapid automated AI analysis of CT scans in neurologic emergencies.
Right, like for stroke.
Yeah,
Ah.
that represents a tangible, potentially immediate benefit of applying this kind of precision medicine thinking using AI to a critical care scenario, getting the right diagnosis faster, potentially enabling quicker, more targeted treatment based on AI analysis of an image.
Yeah.
That's a clear potential win for patients in those very time-sensitive situations.
Okay, so to maybe try and summarize our deep dive here, we've seen how AI is really stepping up to tackle this truly vast and complex world of biological data, genomics, transcriptomics, proteomics, metabolomics.
Hmm.
It's not just analyzing them, but integrating these layers and also applying its power to analyze crucial medical images in radiomics and even those digital pathology slides in pathomics. The core insight seems to be that this combination is building a far more granular, much more personalized picture of disease. This allows for more precise diagnosis, discovering better biomarkers, and optimizing therapies, particularly in cancer, that's clear, but with potential right across medicine, even into urgent care.
And we've definitely highlighted that while the science is advancing incredibly rapidly, there are significant, often intertwined challenges that remain. Wrangling the sheer scale of the data, making it accessible, making it usable, that's still a bottleneck.
Yeah.
Navigating the complex and still evolving regulatory landscape requires clarity, collaboration, maybe adaptation, and figuring out the right economic models and securing that crucial payer adoption is absolutely vital for widespread implementation.
And layered underneath all of that are the equally important considerations around things like ensuring data privacy, ensuring equitable access to these advanced therapies and diagnostics don't just benefit a few, and making sure our ethical practices keep pace with the technological capabilities.
Absolutely. Making the full promise of precision medicine a reality is definitely not something any single researcher or company or regulator can achieve alone. It really requires deep coordination and collaboration between all the players involved,
Yeah.
the scientists pushing the boundaries, the clinicians translating it into actual patient care, the industry developing the tools and therapies, the regulators ensuring safety and efficacy, and the payers who ultimately enable access to it all.
It really makes you think, who stands to gain the most if we fully realize this kind of fundamental transformation of healthcare? If treatment truly becomes personalized, tailored down to the individual level for every single person, who benefits the most? And I wonder, does that make them potentially the most powerful force driving these changes forward in the future. What will healthcare actually look like when or if we finally get there?